Okay, let's jump right in. For a long time, understanding the brain has been a bit like looking at a wiring diagram, right?
Mhm.
We focus on mapping all the physical connections between neurons, the connectome. But the big question is, does knowing where all the wires go really tell you how the machine works?
Exactly. It's that classic structure versus function question.
So today we're diving into a paper from Nature that tackles this head-on, using the little worm C. elegans. Our mission basically is to figure out why just mapping wires isn't enough, what they found when they actually measured the signals, and, well, what that might mean for how we think about any brain.
Sounds good. Yeah, it's a fascinating study because C. elegans has this incredibly detailed anatomical map, maybe the most complete one we have, but function. But function builds on that map, but it's not the whole story.
Right, so let's unpack the traditional way first.
Yeah.
The connectome, that's all about the physical links,
Mhm.
the synapses, yeah.
Yeah, the physical touch points.
And the idea seems solid.
Uh-huh.
Figure out the inputs and outputs and you understand the neuron's job. Feels like having the blueprint.
It does, and honestly, the worm connectome has been invaluable, no doubt about it. We've learned tons about specific circuits, but just looking at anatomy has its limits.
Like what?
Well, for starters, the map doesn't always tell you if a connection is, excitatory go or inhibitory stop. Some neurons can even release both types of signals.
Both. How does that work?
Yeah, like starburst amacrine cells in our retinas, they use GABA and acetylcholine. It often depends on the receptor on the other side or the context. Anatomy alone doesn't easily capture that nuance.
Right.
And it also doesn't tell you how strong a connection is or how fast the signal travels. Plus, and this is a big one, it totally misses communication that happens outside of those direct physical connections.
Outside, you mean like wireless signaling?
Exactly, like broadcasting signals instead of just sending them down a dedicated wire. Anatomy doesn't see that.
Wow. Okay, so even with a perfect map of every wire, you might not know the signal's polarity, its strength, its speed, or about this whole other wireless network.
That's a good way to put it. The anatomy is the infrastructure, the potential roads, but not the actual traffic, the speed limits, or the radio broadcasts happening alongside.
So how did this paper get around that? What did they actually measure instead of just mapping?
They took a really different approach. Instead of inferring function from structure, they measured the signal propagation directly. They looked at the effect of one neuron firing on others.
Huh?
They used optogenetics, basically using light to turn specific neurons on. They engineered the worms so they could shine purple light on just one neuron at a time to activate it.
Yeah.
And while that one neuron was active, they used another tool, GCAM P6s, to record calcium levels in pretty much all the other neurons in the worm's head simultaneously.
Calcium. That tells you
Yeah.
when a neuron is active.
Right. Right. Calcium rushes in when a neuron fires and gets excited. So they're essentially stimulating neuron A and watching to see which other neurons B, C, D light up in response.
And they did this for how many pairs?
Systematically, they covered almost every possible pair in the head. That was, let's see, 23,433 pairs. It covered 99% of the head neurons, and they did this across 113 different worms. So a huge data set.
Wow.
And the result is what they call a functional atlas. It's a map showing if there's a connection, whether it's excitatory or inhibitory, how strong it is, the timing, basically the actual causal influence neuron A has on neuron B.
Okay, this feels like the crucial bit. They're not guessing function from the wires. They're seeing the function directly and mapping that.
Exactly. That's the key distinction. And when they compared this functional map to what you'd predict based only on the known anatomy and standard synapse models, well, the two maps looked pretty different.
Different.
So they found about 1,300 pairs that were functionally connected based on their measurements, a pretty strict definition. About 11% of those were inhibitory. Now, sure, if two neurons had a direct wire between them anatomically, they were more likely to be functionally connected. That makes sense.
Okay, expected.
But, and this was surprising, a really large number of pairs that do have direct physical synapses in the connectome showed little or no functional connection in their experiment.
Wait, really? The wire's there, but no signal gets through reliably.
Apparently so, or at least not strongly enough to be detected under these conditions. And the flip side was also true. They found functional connections, real signal transmission, between neurons that anatomy predicted would have weak or even no connection.
Huh.
And these functional links often span longer paths in the wiring diagram on average. The signal seemed to travel over 2 hops anatomically.
So not just direct neighbors talking.
Right. And when they tried to use the anatomy-based models to predict the functional responses they actually measured, the models did, well, terribly.
Terribly how?
The paper reports an R-squared value of less than 0, which, for anyone who deals with stats, basically means the anatomical model was worse at predicting the outcome than just guessing the average.
Worse than
Dang.
guessing. That's
Wait,
dramatic. So the anatomical blueprint is seriously incomplete then. What's the big missing piece?
The main culprit they identified is that wireless communication we mentioned, extrasynaptic signaling.
Okay, tell me more about that.
It's where neurons release chemicals, often things called neuropeptides or monoamines, from specific storage units called dense core vesicles. But instead of releasing them right across a tiny gap, a synapse, to a specific target,
Yeah.
they release them into the space around the synapse. These chemicals can then diffuse, travel a bit further, and activate receptors on neurons that aren't directly wired neighbors. It's like broadcasting a signal instead of making a direct call. Anatomy doesn't map these broadcasts.
Right, because there's no physical wire for it. Okay, that makes sense. But how did they prove that this extrasynaptic stuff was the reason for the mismatch?
They did a really clever control experiment. They repeated the whole process, activating neurons, measuring responses, but this time in worms with a mutation in a gene called UNC31.
And UNC31 does what?
It's crucial for releasing those neuropeptides and things from the dense core vesicles. So UNC31 mutant worms are basically defective in this type of extrasynaptic wireless communication.
Ah, okay, so they turned off the wireless network, essentially.
Pretty much. And what they found was striking. First, the UNC31 mutants had significantly fewer functional connections overall compared to the normal wild-type worms.
Less communication happening.
Exactly. And second, the agreement between the anatomical predictions and the measured functional connections was actually better in these mutants.
Better. Wait, so taking away the wireless part made the anatomical map more accurate.
Yes, which strongly suggests that it's precisely this extrasynaptic signaling in normal worms that causes a lot of the functional connections that the anatomy map just doesn't predict. It's the missing layer.
That's really strong evidence. So this wireless network isn't just some minor detail. It's a major player in how the worm's nervous system actually communicates.
Yeah.
Do they have specific examples?
Oh, yeah, several good ones. Take a neuron called RID. Anatomy suggests it has pretty weak connections to neurons like URX, ADL, and AWB. You wouldn't expect much interaction.
Tell me.
But in normal worms, activating RID caused clear, strong responses in those other neurons. Then, in the UNC31 mutants, the ones without the good wireless, those responses were much smaller or just gone.
Wow.
And they even checked the gene expression data. RID makes certain neuropeptides, and guess what? URX, ADL, and AWB make the receptors for those peptides. So the molecular machinery is there, even without a direct wire. Classic wireless.
That's cool. Any others?
Yeah, another pair, M3L and URYVL. The connectome shows no direct synapse, chemical or electrical, between them. 0 wires. None. But activating M3L caused a definite response in URYVL in normal worms. And again, that response vanished in the UNC31 mutants. And they found peptide receptor matches in the genes too, things like FLP4 peptide and its receptor MPR4.
So communication happening with literally no wires shown on the map.
Exactly. They even saw it between pairs on opposite sides of the worm's head, like AVDR and AVDL. Weak or no direct connections anatomically, but activating one caused a robust response in the other, and it depended on UNC31. This fits with other recent ideas about these neurons using peptides to talk to themselves and their neighbors.
And didn't they specifically look for pairs that only use this wireless route?
They did. They screened their data and found 53 pairs that seemed functionally connected in the normal worms, but not in the UNC31 mutants, suggesting their communication is purely or almost purely extrasynaptic.
So a significant number relied just on the broadcast.
Seems so. And interestingly, they found this extrasynaptic signaling isn't always slow and modulatory, which is kind of the textbook view sometimes. In the worm, it can actually happen quite quickly, sometimes on timescales similar to direct synaptic transmission.
So it can be fast and wireless. That adds another layer of complexity anatomy misses.
Totally. It really changes how you have to think about information flow.
Okay, so they've got this functional map, including wired and wireless effects.
Yeah.
Does it actually help predict what the worm's brain does normally, like the ultimate test?
Right, that's the key question. Does this map help understand real brain dynamics? So they tested different models to see how well they could predict the patterns of spontaneous neural activity Just the normal chatter happening in the worm's brain when it's doing its thing.
And how do the different maps compare?
Well, models based just on simple anatomical connections, like synapse counts, they were pretty poor predictors of the spontaneous activity.
Okay.
More sophisticated models that tried to account for signals traveling along multiple anatomical paths did a bit better, but still not great.
And their functional map?
The model based directly on their measured functional connections. The one that implicitly includes both the wired effects and all that extrasynaptic stuff, it performs significantly better than any of the anatomy-based models. It was the best predictor of the real correlations they saw in the spontaneous brain activity.
Ah, so that really seals it, doesn't it?
Yeah.
The map reflecting what signals actually do, including the wireless part anatomy misses, is the map that best captures how the network actually behaves.
I think so. Because it's built directly from measurements, it avoids a lot of the assumptions you have to make when you start with anatomy and try to infer function, and it explicitly captures that extrasynaptic component. It just seems to be a more accurate representation of the effective connectivity.
Okay, let's zoom out then. What does this deep dive into this tiny worm's brain mean for us, for you listening? Why is this important beyond C. elegans?
Well, I think the really big takeaway is that knowing the anatomical wiring diagram, the connectome, is incredibly useful, but it's fundamentally incomplete, even in a nervous system as relatively simple as the worm's.
It's not the whole picture.
Not by a long shot, it seems. To really get at how neural circuits function and to predict their activity, you need to actually measure how signals propagate. And you absolutely have to account for this extrasynaptic signaling, the wireless communication, which anatomy often overlooks.
Right.
This idea of effective connections, how neuron A influences neuron B through all possible routes, wired and wireless, seems to be a much better guide to understanding network dynamics than just looking at the direct physical links.
So we need functional maps to go alongside the anatomical ones.
Exactly. This study really underscores that need. As we try to model bigger, more complex brains, including potentially our own someday, we'll need both types of information. And finding that fast, wireless signaling is so important in the worm.
Well, it makes you wonder how much of that we might be missing in other brains, too.
It definitely changes the picture. The brain isn't just a neat circuit board. It's maybe more like a circuit board with a bunch of radio transmitters and receivers operating at the same time. That's a pretty good analogy, yeah. And measuring the actual functional impact, the broadcasts included, seems key.
So any final thought that struck you from this?
Yeah, actually, the paper mentioned something interesting. They observed variability in these functional connections they measured, not just between worms, but even across different trials in the same worm.
Variability, so the connections aren't fixed.
It suggests they might not be entirely static, even in the worm. They could change based on the worm's internal state, maybe what it just experienced, context. It raises this idea. Could there be latent connections? Like the anatomical potential is there, the molecules are there, but the functional link only switches on under certain circumstances.
Huh. Like hidden pathways waiting for the right conditions?
Maybe. This functional atlas they built is a huge step forward, but it might just be scratching the surface of an even more dynamic and context-dependent communication system. Lots more to figure out.
Absolutely fascinating. It really shows how much more there is to learn, even from a tiny worm. Well, thanks for joining us on this deep dive.